Today, we are happy to be joined by Espen Rostrup Nakstad, who needs no further introductions. Espen is invited to participate in the dialogue with the researchers in the two first sessions.
If you have questions to the presenters, please use the chat and we will try to do our best to forward them to the speakers. We also encourage you to use the tools available in the conference portal by, for example, contacting people you would like to meet afterwards. Well, let's start and welcome the first presenter. That is Ludvig André Munthe. He is a professor at the Institute of Clinical Medicine at the University of Oslo and the director of KG Jepsen Center for B-cell malignancies. Ludvig, the stage is yours.
Hello, everyone. Let's see if we can get the slide up, Mikkel. I'm grateful for the opportunity to present the work. Right now, I don't see my presentation, but it's coming soon, I guess. So there we go. I just have one single slide, and I thought that would be best just to give a slight overview for everyone, and then anyone that wants more information can either discuss with me or listen to the second part. Now, the title that you can see at the top there is amended to show more what we are doing right now. So the title is Pathogenesis, Immune Functions and Memory in Mild, Severe and Life-Threatening COVID-19 and in Vaccinated High-Risk Groups. We have been working with high dimensionality analyses. That means mass cytometry, flow cytometry, and other things, and also specificity analyses to look at immune responses in both severe and mild COVID-19. This work actually started because of a worry that we were entering a pandemic that was sort of the big one, a completely new virus not experienced before, that we would have really big problems evading and with high mortality and disease. But it turns out that this is not the case, luckily. The virus is quite similar to the seasonal coronaviruses, and this is the second point on the list there. There's a robust pre-existing cross-reactive T-cell immunity to the SARS-CoV-2 infection, and the T-cell responses is significantly higher, this recall response, than the de novo responses. In addition, when we have analyzed the imprint of this new virus strain compared to the rest, to the other seasonal coronaviruses, we find that there's a common peptide pool for human coronaviruses, and that's part of the analysis that we have been proceeding with. The next part is a project that is funded by an RCN, so Research Council BIA grant, together with NEC on Community, which is a company that has developed an immune profiler that predicts T-cell epitopes in cancer. So this was leveraged to analyze immunodominant epitopes in SARS-CoV-2 virus. We are on the way of using this immune profiler to identify immunodominant epitopes and then also validate this AI prediction platform. At the same time, we're doing extensive biobanking and analysis of COVID-19 patients, both from the severe ICU wards and from hospital wards, and also convalescents. In addition, there we have blood bank donors that represent then negative control subsets, people that have been exposed to the seasonal coronaviruses throughout their previous life. We also are embarking on analyses of T-cell responses to mutants, so these new viruses, and have ordered 700 peptides to investigate immune evasion mechanisms. Also, the biobanking analysis has progressed to look at vaccinated, so the vaccination program, which is the next major part of what we're doing. We're biobanking before and after samples, both serum and cells, from a large number of patients and controls. And we've also sent a national application to an organization called CEPI on behalf of many organizations. So it's Oslo University Hospital, NIPH, Diakonhjemmet, Ahus Sykehus Østfold, and also ClinMed of University of Oslo. This is a major project and it will take some time to describe. You can hear a little bit more in the next session. Last, we're also involved with analyzing adverse effects from the AstraZeneca vaccine. So that was a brief overview from me. Last, funders, we are a University of Oslo lab. Kreftforeningen, the Cancer Society, funds much of our work. In addition, Oslo University Hospital has funded this. In addition, of course, NIPH is a very strong partner in all of this work.
Thank you very much, Ludvig. And as you said, we're going to meet you both in a parallel session, but also when we meet in a panel discussion very soon. The next presenter is Kristin Hofsø. Hofsø is an experienced intensive care nurse and is currently a researcher at the Oslo University Hospital and an associate professor at Lovisenberg University College. Kristin, the stage is yours.
So, thank you. I do not see my presentation. Okay. Thank you for inviting me to present our study. This is a study of the Norwegian cohort of patients admitted to ICU because of COVID-19 during the first year of the pandemic. As we all remember, in the beginning of January last year, China announced the identification of the new coronavirus as the causative agent for an outbreak of pneumonia that often required ICU treatment. Okay, thank you. So the main cause of ICU admission was early described as acute respiratory failure with the majority meeting ARDS criteria, which is an even more severe form of respiratory failure and is associated with both high mortality and long-term health challenges related to both mental, cognitive, and physical health. The first reports of ICU mortality with the new coronavirus disease were estimated to be between 49 to 67%, and that up to 22 of hospitalized patients would require ICU treatment. Therefore, it was an urgent need to investigate the new ICU population in a national observational study to identify both mortality rates, patient characteristics, and long-term outcomes. The combination of knowledge from similar patient populations, early reports, and a national register of all intensive care patients in Norway gave us the opportunity to conduct this study. We have published the results from the first wave of the pandemic in Norway, which was from the first ICU admission on the 10th of March last year and until the end of June last year. The age distribution during the first wave had a median of 63 years, meaning that half of the population were actually less than 63 years. The ICU mortality in this Norwegian cohort was only 18%, which is much lower than both results from early reports in the pandemic, but also compared to other patient populations with acute respiratory failure. And the highest risk factor for death was age above 65, as you can see here with the red line. When it comes to the long-term effects of the disease, as fatigue, cognitive deficits, and shortness of breath, We are still collecting data and have just started to look into some preliminary results. We expect that several of these patients will have health challenges at both 6 and 12 months. What we don't know yet is if these health challenges actually are worse, persist for longer, or differ in character compared to other patients treated in the ICU for acute respiratory failure. But we need to be patient so that we can summarize these data correctly and that we don't stress the preliminary results too much in the meantime, as they can be biased. So, thank you. That was my short presentation.
Thank you very much, Kristin. The next presenter is Rebecca Cox. Cox is a professor in medical virology and head of the Influenza Center at the University of Bergen and Haukeland University Hospital. Rebecca, the stage is yours.
Thank you very much, and thank you to the Research Council and the Trond Mohn Stiftelse for the funding for this project. At the beginning of the pandemic, we set out to follow what the whole pandemic in Western Norway. So we followed healthcare workers and the general population. And here on the right, you can see the team that set up this. This is myself, Nina Langeland, Bjørn Blomberg, Carl Brogstad, and Kristin Mohn. And on the left, you see our medical students who have been fantastic at ensuring that we have managed to test everybody in Bergen. So the way we approached this was we knew from the swine flu pandemic that a lot of the healthcare workers were exposed and didn't know it. And we could show this using antibodies during the swine flu pandemic. So we used these yellow Y-shaped antibodies to look at infection rates. Initially, on the right-hand side, you can see that we looked at household infection rates. Unfortunately, we lost the colours, but you can see that 16 people that were PCR tested in a household setting had antibodies, but did not come up PCR positive. So we were able to show that in a household setting, and if you want to know more, follow us in the next session, that antibodies were a better predictor than with PCR, the two of them together could predict infection, showing the importance of infection in a household setting and importance of isolation. Furthermore, we have now looked at the long-term complications at 6 months, and this is by age group. And if we look on the right-hand side with the red, we see that the young people under 15, they have long-term complications of taste and smell, but they do not seem to suffer any other complications. Unfortunately, and the majority of our cohort are community isolated patients, we see problems with memory, concentration and dyspnea. So the impact of COVID-19, people used to say it's just an influenza. This virus is not just an influenza. It is very severe, and the long-term complications are particularly severe, and we see this in ages from 16 upwards. And I think this is why it's really important that we ensure that people get vaccinated. When we looked at the antibodies, we see with time that with severity of disease that the antibodies increase, and we would expect that the hospitalized patients that have high virus load would of course have higher antibody levels. But we see also very high levels of antibodies in the community cases, and we see quite a large number of asymptomatic cases. We also looked at the risks of infection in healthcare workers, and unfortunately we've seen that many healthcare workers have succumbed during the pandemic. And we were able to show in our first paper of 625 healthcare workers using serology that 12 of these 625 healthcare workers were positive, and we found that 4 were asymptomatic, so possibly being able to spread virus in the hospital without knowing that they had the symptoms, particularly in the first phase. So I've got lots of exciting results to tell you a little bit later, but I'd just like to summarise by thanking everybody. We've had a fantastic team. The dugnad word from Norwegian word really summarises. Everybody has been really, really helpful. We've had Haukeland University Hospital, the University of Bergen, Stavanger University Hospital, Bergen Kommune and Haraldsplass Hospital that have been involved. And here are some of the people. So thank you very much.
Thank you very much, Rebecca. Then I would like to invite all the 3 speakers and Espen on the screen. Espen, you have been listening to the 3 scientists. What are your immediate reflections?
Well, my immediate reflections is that this is very important research, and it's the kind of knowledge that we actually need now in this phase of the pandemic and also in the foreseeable future. And I think Professor Munthe was very spot on the variety of analysis on the immune responses in severe and mild COVID-19. That's exactly what we need to know more about, because it's key to understand the disease, it's key to understand how to treat patients, and also to understand the potential side effects of vaccines, for instance. So I mean, it's really a hot topic. And I also think it was interesting to hear the survival rates and long-term outcomes of COVID-19 patients in the ICUs, because I have worked at the ICUs for many years and I know how complicated it is. And as regards to the mortality rates, for instance, why is it so that the mortality in the ICU was about 18% in Norway, whereas in some ICUs in New York in April of 2020, it was 80%. And that has to do, of course, with the momentum of the stress factor really involved also, not the disease itself. So it's really important to have knowledge to improve the care of ICU patients with COVID-19, and to also work, of course, in a structured way to improve outcomes. And I also know, of course, the research of Rebecca, which I think is absolutely what we need now. We need to understand more about the long COVID. We need to understand why young people are affected, how they are affected, and for how long can they anticipate to have these symptoms. And so, I mean, this is spot on, and it's research that is needed and wanted all over the world. So I think it's good to see that scientists in Norway are so eager to contribute.
Let me follow up. Sorry, Kristin, Espen mentioned that it was 80% mortality rate in New York, whereas you presented like 18%. How come? What's the reason for that enormous difference?
I think Espen already mentioned some elements of it. Like in the beginning, we had to do with we had a pandemic in the society that we didn't know how to respond to. We also had a lack of equipment, and we were overwhelmed with ICU admissions, the numbers. But and I think some of the data are quite skewed as well, that it's not really representative data. But also I think there's one still, when we adjust for all of this and look at newer publication on ICU mortality, we still have very good results in Norway. And I think it's important to acknowledge that we had a healthcare system that were still working, and that the one who gave the ICU treatment were actually healthcare workers that normally work in the ICU setting. I think that is of very much importance in this. And of course, it's a lot of different elements into this also, like the but I think that is a very important aspect of it, because the ones who gave the care to these patients, as I said in the introduction, acute respiratory failure is something that the ICU are used to treating. The difference here was that it was a new virus, and the patients came in rapidly in big numbers. But I think that is important, and that is important when we discuss the preparedness for a pandemic due to the capacity of ICUs or ICU treatment.
Espen mentioned the importance of basic research. Roughly 1 year ago, we all responded extremely fast. You got only a 10-day notice to file your application. And we got more than 120 applications within that 10-day period. It was almost like you were prepared. You were sitting there waiting to apply. However, you were not informed. In what way do you think that researchers doing groundbreaking science should be considered part of our defense system against future unknown threats? I could start with you, Ludvig, being a typical basic scientist. What should your role be in a crisis like this?
I'm quite confident that research-driven knowledge is really essential in such crises. And to be honest, I think we as a nation had really poor preparedness to leverage that knowledge base and to resources for such research. But the first call, this one we're talking about now, was sort of the first emergency opportunity to let the researchers apply and share some of their knowledge and technology to help us in this crisis. And I think that our neighboring countries had much better funding schemes and much higher. But anyway, I mean, it has turned out to be quite okay in the long run. We've had, for example, in Oslo, so far analyzed 34,000 sera. This is work by Fridtjof Lund Johansen's group with technology that he has developed. And 34,000 sera, I mean, that's quite a lot. And not so much funded, actually. So it's what can you do with motivation and highly skilled staff, as an example. The same goes for many of the other projects that are around in the region here in Oslo, I think. But I think there should be a funds for emergencies like this, both for biobanking in the hospital setting, so in the health regions, and also for emergency research that we need. It's not only personal protection equipment that we need in such crisis, it's how to turn around and face the challenges we have.
We are living in a time where scientific data make headlines, I would say, every day. And I also, at least from my perspective, scientists are the ones that lead the way out of the pandemic by providing knowledge about the virus, the disease, and of course the vaccines that we all are reading and hoping for. The new results are being published almost as fast as the data is generated and without the normal peer review process. And these results are, of course, as we know, being translated into important political decisions immediately. For most part, this, I think, is very good. But are things moving too fast? Kristin, I would like to start with you.
So I think that is a very relevant dilemma for us being scientists in this pandemic, because there's such an urgent need for new evidence and new knowledge, and the world and everyone is just eager for more information. And the journalists have want to present new results rapidly. And I think it's important. It's a dilemma because we also need to be sure that we don't publish too preliminary results, because they can be skewed and that would not make them generalizable. And also we can have issue with sort of double publication and all of those issues as well. Still, it's our duty to publish new results as soon as possible, because we need new evidence. But I think it's a bit difficult because I'm holding back presenting our preliminary results just because I'm afraid it's going to give a wrong picture of how especially when it comes to long-term outcomes for the ICU patients, because we need to look at it in robust data to be able to see whether this is So I think that is difficult, and I think as a researcher and a scientist, you have a much bigger responsibility than you do as a journalist.
Espen, you are in the midst of all of this, giving political or scientific advice to the government. How do you use scientific data when you give advice, knowing the fact that it's very early days and things are moving extremely fast?
But first of all, I can say that it's very hard to do evidence-based crisis management during a pandemic to a full extent. It's impossible. We can't wait for all the evidence. We would be 2 years behind. So we have to make use of what we have, and we have to see it in a broader context. And from my and the Directorate of Health perspective, we have followed every development in every country, starting from the Hubei province in China more than a year ago, to learn as much as possible, to link this to available research, even though it's not published, and to try to connect the dots to the extent possible to sort of get some kind of sense out of it. And it's really hard. And I think one of the reasons why this crisis has been managed differently in different countries is not only political differences. It also has to do with the element of, or the level of evidence-based crisis management. If you really want to wait for all the evidence, then, as I said, you will be behind. But still, it's not very smart to ignore all the preliminary evidence because it could be very valuable. So it's a very tough balance, and my only message is that we need all the research we can get. We need to treat it with caution. We need to use the principles that we learned as scientists to be careful. And I think that eventually will help us also now in the really tricky questions we're facing now with the side effects of vaccines and other issues. I think we need to use the same principles in the approach to those problems.
You mentioned vaccines. We have developed vaccines faster than ever, and today we are talking about developing vaccines in less than 100 days. Rebecca, you have been working with influenza vaccines for years. Why have we succeeded in making a COVID-19 vaccines, and to what extent have your own work contributed to this?
Yeah, thank you for that question. I think that we have succeeded this time because we've built upon past failures. Going back to having problems with HIV, but also because we've worked extensively on pandemic influenza and developing platforms that can rapidly respond, such as messenger RNA, such as the adenovirus vectored vaccines. So a lot of this is based on the experience we have. In some ways, we were lucky, if one can say that we had Middle Eastern coronavirus, because a lot of the structures and the expertise in how to make coronavirus has been developed. And of course, CEPI has played a central role in this. The work that we've done here has contributed. Firstly, the Novavax, the adjuvant in the Novavax was first tested in a phase 1 trial. Actually, 2009, we were doing a bird flu trial before the swine flu came, and that adjuvant is now going to be licensed in the Novavax vaccine. And we also have worked on nanotechnology with RNA vaccines for flu for many years. So a lot of the expertise that we have needed has been available within influenza, within the Middle Eastern coronavirus field, and I think with funding from organisations like CEPI and NIH, it has ensured that we could rapidly develop these vaccines.
These days we are experiencing vaccine side effects of the most serious kind. First to you, Espen, and thereafter to Ludvig. To what degree do you think that the side effects that we experience will impact the trust to the system and the people's willingness to be vaccinated? And of course, you, Ludvig, being an expert in immunology, can you shed light on what we are experiencing these days? Espen first.
Well, first of all, I think it's extremely important to be transparent and to be honest when it comes to these potential side effects and what we know so far. I think as regards trust in the population, I think the worst thing you can do is to try to hide knowledge and not be transparent. I think that could have a huge impact on people's willingness to actually be vaccinated, regardless of type of vaccine. I think the level of trust goes for all the vaccines that we have at hand. So transparency is key, and I think so far our national authorities have been honest and open to the extent possible. But I think this is an issue all over the world, because the potential consequences of decisions in nations regarding this vaccine could affect also the use of the vaccine in other countries. So it's a really tough dilemma. It's really tricky questions and hard decisions, and I think the only way to do it is to be transparent and open.
Ludvig, will you add?
I think that vaccines are really important for the main reason is to protect the patients that are worst of the worst in a way. And the worst of the worst, the people that have extreme high risk of dying from COVID are the immunosuppressed patients. And on the top, you have patients that are lung transplanted, and then, for example, kidney transplanted. There are 3,500 of those in Norway. These are the ones that we need to protect. These are the ones that we need to make sure that the vaccines work in. They are the ones that are right now actually hiding, are in seclusion to avoid in the infection. So that's the main goal, to protect all the high-risk groups in the vaccination, and also after the vaccination to make sure that they've had a protective immune response. And this is really important, and an area where there is no actual vigilance right now. There's but where we are trying to monitor the immune responses. And internationally, the results there are 15% of organ transplanted get productive immune responses on vaccine. So far we're not even close to protecting these vulnerable groups. The next, when it comes to adverse effects, I think we have to sort them out and publish them and discuss them. And I think personally that it's really good that the AstraZeneca vaccine is on hold, so that we can find out what to do in the next phase.
We have to end this panel discussion with that comment. There is one question and one comment in the chat, and we're going to save those for the parallel session for later. Thank you very much, Rebecca Cox, Kristen Hofsø, and Ludvig Munthe. And as mentioned, if you want to talk to them directly, please join their parallel session. The next presenter is Per Olav Skjervold. Skjervold is a co-founder and chairman of the board of NorImmune. Skjervold, the stage is yours.
Following the pandemic outbreak, our scientists started to ask, could we possibly transfer our technology and use our competence to aid the fight against COVID-19? What we normally do is developing antibody passive immunization treatments against parasites, bacteria, and viral diseases in aquaculture and farmed animals. This veterinarian approach is based on our ability for large volume production of polyclonal IgY, antibodies extracted from eggs, following immunization of the hens. My name is Per Olav Skjervold from the company NorImmune, and we've had a fruitful collaboration with Professor Magnar Bjørås at NTNU and Oslo University Hospital in this project. Our aim is to contribute the fight on the ongoing pandemic by further developing antibody treatments, preferably administered in the respiratory tract and in combination with already approved treatments. Since this project started, last year, we have produced polyclonal antibodies targeting SARS-CoV-2, spike proteins, and other epitopes. What we've shown is that the IgY antibodies give strong in vitro neutralization of wild-type SARS-CoV-2. This is for us a clear proof of concept, and we will now validate this further in animal trials. Using antibodies from convalescent human plasma or as monoclonal antibodies is well documented and used in the absence of vaccines. This combination was already in use with already approved medicines. This was actually the treatment given to former President Trump, and the Lilly monoclonal antibodies is recently approved. We believe we can develop a similar approach based on cost-effective and large volume polyclonal IgY antibodies as a first-line COVID-19 treatment. We believe this could also be part of a long-term risk management strategy fighting future pandemics. Norway has the advantage for IgY production due to the unique good animal health, and our technology for rapid and large volume production can be explored to fight the pandemic. We think we can therefore develop a Norwegian cost-effective alternative to monoclonal antibodies. We need to utilize this potential as a part of a first-line strategy. We are now also looking into the potential benefits of IgY being polyclonal, as monoclonal antibodies and vaccines might lose its efficacy due to virus mutations. So we are now testing the IgY ability also to neutralize new mutants of the SARS-CoV-2, and we'll also investigate if we can even strengthen this by giving more robustness through a more complex mixture of antibodies. Okay, we look forward to a more in-depth presentation and a discussion with you later. Thank you.
Thank you, Per Olav. The next presenter is Catherine Hadley. Hadley is Head of Clinical Development in the company Agelab. Catherine, the stage is yours.
Thank you, Ole Johan. It's great to be here today. There we go. Agelabs is an Oslo-based diagnostics company that develops biomarkers using machine learning on large epigenetic datasets. The aim is to detect age-related diseases earlier. We have teamed up with the Norwegian Institute of Public Health, Oslo University Hospital, and Bærum Hospital to develop diagnostics for COVID-19. Our joint project called Predict COVID, or PredCOV, has received funding from the Research Council, for which we are very grateful. Our goal in the project is to develop diagnostics for predicting who will have a severe outcome early on, enabling earlier treatment and hopefully better outcomes. We are also looking into long COVID, which is the focus on this in this presentation. Our method is to analyze the switches that determine which genes are turned on and off in the immune system in white blood cells. This is also called DNA methylation. These changes often precede other conventional biomarkers, making it possible to detect signals much earlier. Also, these switches may change in response to environmental factors such as exposure to viruses. We have just started a data analysis on our first data set on post-COVID. These are samples taken 6 to 12 weeks after COVID-19 infection, and we got some help biobanking the first sample. Our main finding is that there are significant differences in gene regulation in white blood cells that can be detected 6 to 12 weeks after infection with COVID-19, and these changes correlate with disease severity. Our findings suggest that COVID-19 induces changes in gene regulation, and these changes last several weeks after an infection. One of the genes that are affected is known to be involved in a wide range of autoimmune diseases, including SLE and Kawasaki's disease. It still remains to be seen whether these changes are transient or if they may trigger autoimmune diseases in some patients later on. Our next steps will be to analyze long COVID to see if there are methylation changes in certain genes that correlate and may explain the tiredness and memory loss experienced by 10 to 30% of patients after COVID-19. Let me see. So if you're interested in collaborating or hearing more about our project, please reach out. I want to thank our fantastic PredCOV team, and thank you so much for the attention.
Thank you, Catherine. Then I would like to invite Per Olav, Espen, and Catherine to a panel conversation. Again, Espen, I would like to start with you. You've been listening to these two last presentations. Immediate reflections?
Well, same reflections as in the last session. I think this is very important research, and I think the question of antibodies, and I mean ever since we saw Trump on TV, I mean how fast he recovered against all odds, actually. I think that has been one of the treatment regimes that actually has proved successful, in contrast to all the previous tests of existing medication that hasn't really proven to be very effective. So I think this is at least from a clinical perspective, really important research to that might actually lead to improved treatment, also not only in the ICUs but also in after hospital admission in general, to prevent patients from needing ICU treatment. And I also think catchment research is really promising, and it's interesting, and I think it's only the first start, of course, of this research, and I hope also that this has spillover effects to potential future pandemics and other parts of infectious diseases, not only the SARS-CoV-2 virus, because this could actually be platforms of further improvement in future healthcare challenges, of course.
Per-Olav, as mentioned, the former President Trump, during the last year, there has been very much focus on vaccines, and we have seen, as we mentioned, new vaccines have been developed very, very fast. How does your strategy by developing polyclonal antibodies fit together with that, and how does it fit in a situation where the virus is changing, not all the time, but you see new mutations coming up? Per-Olav.
Yeah, thank you. So as I mentioned, our background is that we worked on these similar questions for animal and aquaculture species where vaccines is not available. And we from that, we know that this works. And our approach has then been to do this in a large volume compared to when you use antibodies for diagnostics, it's small volumes. We are talking tons. So what we have experienced is that we know we can very rapidly do this. When we got this idea, it took 5 to 7 weeks, and we had large volumes of polyclonal IgY that we could start to test with Professor Magnus Bjørdaas' group. So I think it has the strength of rapid volume production that is very different from what we see with the monoclonals. I often compare if monoclonal is a rifle shot, it's strong if it hits the target. But me being a hunter, I use a shotgun because you have a higher probability of hitting the target if it's moving. And that's another thing that we haven't seen that results yet, but there's good reason to think that this is a part of the polyclonal antibodies we are looking into. So it's I really, of course, this is mainly privately funded based on our like everyone else, we would see if we can help. But I think further discussions with the authorities on how we could be part of this strategy for future pandemics is also extremely important for us.
The same question to you, Catherine. Your system is based on gene responses, and how do you expect the gene responses or the epigenetic changes to be affected by the various types of the virus as they develop?
That is a good question, and we don't know yet. The benefit with software-based diagnostics is that you can continuously update it and continuously improve diagnostics based on real-time data. So as for now, we still include patients, so we have the possibility of improving as we go.
Per-Olav, back to you. How do you see your strategy compared to the reconvalescence serum, the monoclonal antibody strategies, and we have seen drugs being approved? And if it is so successful or so potent, why haven't we seen it been applied anywhere in the world if it hasn't?
Also, you mean the polyclonal? Well, I've been working with innovation in many areas, and I often see that it's of course, as you know, when you mix different disciplines, that's when things come up. And I think that's the background for us, that we have worked in a totally different approach on large volume production. And I think that is, the benefit. It's more than, I think, 1,400 manuscripts showing that this works fighting virus, that IgY. and the strength of IgY being further from IgG, so you have less risk of activating the immune system that is not desired. But it has been maybe a question of production cost and scaling this up. And that's a question that is not relevant for us because that's where we come from. So I think that's mainly the main idea, that we saw this idea because production costs should not be the limitation. Of course, I mean, there's a long way from us to go into we have our network in aquaculture and veterinary medicine, but to establish the platform for GMP production in Norway is, of course, something that we don't have that network. So that's one of the reasons why we are here. Previously we've been not very active in communicating what we're doing because we have the network, but here we don't have the network. So,
Per Olav, you mentioned two keywords. You mentioned production and you mentioned Norway. I'd like to go back to you, Espen. Quite a few people are talking about that we should produce more things in Norway, more drugs, vaccines, medical devices, protective gear, and so on. How do you see us being even better prepared for the next crisis by increasing the amount of production that we have in the health sector in Norway? Espen.
Well, this is not only a question of the preparedness or the level of preparedness in Norway, but also in the Nordic countries and in Europe, because as you know, this is being now discussed all over Europe. How can we reduce our dependency on Asia, for instance, when it comes to drugs and also vaccine production? Although we do have plants in Europe and US that produce vaccines, of course. But I think this is a political, health political issue, and it's being debated and discussed. I don't believe the answer is that we should produce everything in Norway. I think it's impossible, as you all know, to scale up to that level in a small nation like Norway. But cooperation with neighboring countries is one solution, and also a European joint effort to strengthen preparedness in all these aspects is, of course, key now and vital. And in the foreseeable future, we know that there will be more pandemics. It's not going to be 100 years till next time, that's for sure. So this is a momentum now to have these discussions and to join political effort and also science and the entire health sector, basically, to come up with good solutions that are sustainable and that has a future, actually, both commercially, of course, and also for the health sector.
To follow up on that, we have, as we all know, a defense and preparedness system for the seasonal flu. Do you, Espen, and the two others, do you see that we should develop a kind of a pandemic preparedness system or a defense system, meaning that we should do a lot more testing, a lot more sequencing, and that means both in people being hospitalized with an infectious disease, but also in the environment, birds and other animals, and share that information very quickly across borders, and then have a system to develop the vaccines very, very fast. And that, at least from a technological optimistic point of view, that could actually make us very well prepared in the sense that we should not see a new pandemic. Espen first.
Well, I think you're into something really important. I mean, how can we scale up testing and tracing and all the other things that are important in the initial phases of an epidemic to avoid it from being a pandemic? And this is an area where I think that some nations will actually establish good systems that at least are better than what we had. But I'm not sure that the entire WHO system and all the member states will step up to the same level. I fear that in many countries, once this pandemic is over, whenever it is over, of course, that one would easily fall back to old systems and not really spend too much resources on rapid testing and tracing capacities when the need is not there on a daily basis. So it will be interesting to see, and I think the question is really good.
Per Olav, Catherine?
I'm afraid of going outside my expertise, but I would just mention, I mean, we come from the veterinarian side, and at least I've seen that the it's already platforms for large screening of production animals. That I think, so I think they had for the health, also the medical community to look into the veterinarian practices, because they've been doing this for years. So maybe there's something to learn.
Catherine?
One thing is the screening and surveillance, but I was hoping also it would be possible to have a pandemic research preparedness, that there are studies and protocols in place, so it would be rapidly able, or they would be rapidly able to start the clinical studies that is necessary to obtain more information.
We have one question from the chat. It's from Marianne Jare. She's writing, preparing for the next pandemic is one thing, but the next crisis might not be a pandemic. So how do we develop preparedness more in general? Could start with you, Espen.
Well, it's another good question, because what we do know for sure that the next health crisis will definitely not be a SARS virus. At least it's not very likely. So it will be either another epidemic or it will be something totally different. And it could be drug resistance and other problems that we will face. So I think it's a good question. I think many of the things we have learned and the preparedness that has been improved in the recent 12 months is something that can last and that can help us have a broader and more solid preparedness for future events or health crisis. Exactly how it should be and what it should look like is a bit too early to tell, but I think it's wise to work on it and to make sure that all the money and resources put into preparedness also has a value on a daily basis. I think that's when it works, when the systems work on a daily basis, but can easily be switched over to preparedness systems when needed.
Catherine? Do you want to add something, Catherine, to this topic? No? Then I would like to, one final question to you, Catherine. As mentioned that hopefully we will see the end of the COVID pandemic. However, some of the patients, they experience long-term complications. As you develop your test, do you see that it can pick out the people in especially a high risk of developing long-term complications?
This is too early to say certain, but we have started the analysis on long COVID, which I think is interesting. And I'm afraid we're just seeing the tip of the iceberg when it comes to long-term effects. If you look at the Corona study, the results they have so far, which show that 30% of patients who've gone through COVID, they report long-term effects 3 to 6 months after the infection, and this is regardless of age and disease severity. I find it quite concerning.
Thank you. Per Olav, I see you raised your hand, but due to time, we have to stop there. So thank you very much, Per Olav Skjervold and Catherine Hadley. And if you want to talk to them directly, please join their parallel session shortly. Then I'm going to give the word to Sveinung Hole, who will chair the next session. Sveinung.
Thank you very much, Ole Johan, and thank you very much to Espen for finding the time to attend. Espen, we really applaud your contributions in communicating science to the public and to all of us in this extraordinary situation we're all in. Thank you very much. So let's move on to the economical consequences. Lasse B. Lien holds a Dr. Econ and Master of Science degree from the Norwegian School of Economics, NHH, and a BA in Political Science from the University of Bergen. He will introduce the project together with Professor Bram Timmermans at NHH. Please, the word is yours.
My name is Lasse B. Lien. This is Bram Timmermans. We are both professors at the Norwegian School of Economics, and we're in charge of the project COVID-19 and Human Capital, Cataclysm and Catalyze. Our project has two overall objectives. First, to study how the pandemic impacts human capital decisions by firms and the economic and labor market consequences that this implies. Secondly, we want to investigate the interaction between COVID and digitalization. The economy, after all, was midstream in a process of digital transformation when this pandemic hit. And to study these questions, we are now in the process of developing a large integrated database that combines detailed national registry data with survey data.
And while we are building this large integrated database, we have started collecting and analyzing some of this survey data. And so far, we have focused our analysis on the innovation response as a reaction to COVID-19. Crisis and innovation are, of course, strongly linked. After all, there's a well-known saying that states that necessity is the mother of invention. And we're curious to identify and investigate whether this also is valid in this particular case. In the media, we've seen examples of companies that innovated vigorously and rapidly. For example, this gin distillery that tweaked their production process to produce hand sanitizers, or, of course, our restaurant around the corner that now suddenly offers takeaway services. And when we study this innovation and innovation responses, we are interested in several dimensions of innovation. First, we are interested in the extent that innovation actually happened. We do so by asking questions like, did you introduce new products and services? Did you have new working processes? Did you enter new markets? And maybe introduce new forms of logistics. Secondly, we also wanted to know the characteristics of firms that actually engage in innovative processes. And finally, we wanted to see the extent that innovation has value when the crisis is over. After all, many of these response, for example, your takeaway service, is maybe was a short-run response rather than a long-term valuable response.
So from our survey, we see that innovation activity has indeed exploded in response to COVID. Corona-driven innovation was zero in February 2020 when the crisis started. But already by May 2020, around 45% of all firms had introduced at least one innovation in response to the crisis. And by the end of 2020, this number had increased to more than 76%. We also find things like that the amount of innovation increases with firm size, and particularly that firms that had capabilities and focus on innovation before the pandemic started tend to do more and do better. We also find it interesting that Corona innovation seems mostly to be useful in the long run. 72% of Norwegian CEOs indicate that they believe that their COVID innovations will be valuable when the crisis is over.
So while we see that innovation activity has increased, there are also some questions that remains open and that we hope to address in the project later on. So first, what actually happened to, let us call it, ordinary innovation activities? Have Corona innovations crowded out such innovations? And if so, what are the long-run implications of this? Second, we're also interested to understand business model implications and how firms have innovated their business model as a response to COVID-19. But again, these are some topics we will discuss in the future.
Thank you for joining us here, and we are looking forward to any comments and remarks that you might have.
Is Erling Barth. Barth is a research professor at the Institute for Social Research in Oslo. Erling, the screen is yours.
All right. So I'll present our project, which is on labor demand in crisis and recovery. What happens to firms' demand for labor during the pandemic and after the pandemic, if we get there? So we're going to track the demand for labor through the different phases of the pandemic and the recovery. So there's the immediate effect on behavior of the spread of this virus, and there's also direct effects of policy measures when the government locks down certain activities. And it's difficult to disentangle the effects of both of these, but that's one of our goals, is to look at both of these impacts and see how important various policies are and how important the responses to the spread of the virus is. And then we move on to study what happens when the pandemic is resolved. And we have which presumably happens when we are vaccinated, and then hopefully happens. And then what are the conditions for the recovery at that when that happens? So one question is, of course, related to the possibility of subsequent crisis. So will we face demand crisis? What will be the consequences of global shocks on the Norwegian economy? Shortages in the global supply chains, drop in demand for countries who do not improve as fast, a possible financial crisis, political unrest, and so on. But we don't really know if this is going to be a V-shaped and quick recovery, or if it's going to linger on, and to what extent and what kind of problems will that be. And another question is related to changes in technology and structural change. So the pandemic may enhance or hamper the speed of technological change in certain directions, and this will affect the types of labor that are needed in the future. So our project has a focus on youth, jobs for youth, and it has a focus on youths are hit more by this pandemic, and the consequences for youth of not getting a job may be long-term consequences. There are scarring effects potentially, and there may be long-term effects from learning, and then once the pandemic is over, they will face the competition from a new generation of young people who are ready to enter the labor market. So it's a pertinent question how the demand for new jobs evolves. So our focus is on new jobs and job creation. One thing is the re-employment, of course, of those who lost their jobs, but our focus is on the new jobs and on the job creations for the future. So we'll first use real-time data on vacancy postings to look at job openings for newcomers and reallocation. Then, as we move on, we will look at realized hires, exits, churning of jobs, and job creation and job destruction in firms, and try to tease out who will be the winners and the losers among workers and firms, and look at effects on inequality and productivity. So the project is an international comparison. We will compare with countries who have followed different strategies. And the question is if this led to similar paths in labor demand. And this is a collaboration with Harvard University, Uppsala University, and Aarhus University. And then we have a collaboration on data and dissemination with the NHO and NAV in Norway. This is a quick peek into what happened with job vacancies. This is the vacancy ratio in 2020 versus 2018 and 19. So we have weeks here on the x-axis, and then we have normalized the job vacancies with the average vacancies from 2018 and 19 in the same week. So the straight line here represents the benchmark, which is the same weeks in 2018 and 2019, and then the black dotted line here is the relative number of job vacancies posted. This is the University of Job Postings in Norway in each week compared to the 2 other years. So we see we had a drop of about 50% in week 15, 16, which was so this is the beginning when the new cases came in week 8 in Norway, and then we had a a lockdown in week 12. I mean, this is when we had the first cases, and then this is the lockdown, 12th of March or something in week 12. And then we see that jobs for youth, this is occupations that are important for youth, for the 20 most important occupations, 3-digit for young people, and they were hit much harder. What we see is that they also improve more as the virus, as we had an improvement in the spread of the virus during the spring, and then it dropped again in the fall, and it drops when the virus basically spreads more again. So one thing, yeah. So this is the start of our project, and I'll close there, and there will be more details in the later session. Thank you.
Thank you very much. The last presenter today is Simen Markussen. Simen is a senior research fellow at the Frisch Center for Economic Research. Simen, the floor is yours.
Thank you so much. I'm here today as a substitute for my colleague Oddbjørn Roem, who is the project manager of this project called the Corona Crisis and its economic consequences. And the starting point or the background for this project is that more or less immediately after the lockdown in March 2020, a bit more than a year ago, we established a cooperation between the Frisch Center, Statistics Norway, and NAV, Norwegian Labour and Welfare Directorate, in order to enable the data to conduct fresh empirical analysis on admin data. So we got hold of data for UI applications, unemployment applications, because that was sort of the most updated data source available to conduct empirical analysis on the crisis, sort of almost in real time. And the ambition of this project is sort of to continue that work, and also to go back and sort of reanalyze the crisis with the data that has been available for research now that was not available back then. And a general point here is that in Norway we do have a quite unique data infrastructure that do enable real-time analysis on a broad range of topics to a much larger extent than what we have today. If you are able to sort of cut red tape and make this data efficiently used. And this project will analyze a broad range of topics, but to sort of summarize the topics, we're going to study unemployment dynamics and labor market dynamics during the crisis and in the aftermath of the recovery process after the crisis. And questions such as how is unemployment durations affected by the different policies? To what extent do we see mismatch in the labor market, job mobility across sectors, entrepreneurship among the unemployed, etc., etc. The project involves several partners. In addition to the Frisch Center, it also involves researchers at NMBU and also in Munich and Copenhagen. And additionally, we have partnered with NAV and the major labor market organizations in NHO and LO. And we aim to have impact sort of on two fronts, both societal, that we believe that these kind of updated but perhaps simple analysis are of some use and importance for policymaking and the public during the crisis, and also to hopefully have some academic impact in order to contribute to the fast-growing body of research and hopefully publish in high-ranking journals. Okay, thank you.
Thank you very much. Then I will ask Lasse, Erling, and Simen, please join me on the front screen. In my simple words, you are studying human capital decisions at company and individual levels. You're studying effects on labor market demands for various groups and subgroups, and you're studying a more total perspective on the crisis and the recovery. Also, all in all, you will provide us with a rich view on the economical consequences of the pandemic. So, having heard your presentations, and to all of you, now that, do you see any aspects of your research projects coming together, complementing each other in any way? Please, Lasse.
Well, I think we're clearly partially overlapping, and I think that's because we're responding to the same stimulus, all of us. One is the call from the Research Council, of course. The other is that all three projects tend to wish to utilize the superior quality of Norwegian registry data to understand the dynamics of the crisis using high frequency, high pixel data. I think in a way we will probably be replicating each other, and perhaps small nuances, but there's quite a bit of overlap. But I'm sure this overlap is not perfect. For instance, it will always seem much, the overlap will seem much bigger when you're presenting in 3 minutes than if you really get down to the details. But from what I saw from the other presentations, I think what's perhaps unique about our research project is that in addition to using the registry data and tracking the dynamics and changes in the economy, we also focus a lot on understanding how the decision makers at the firm level decide, how are different characteristics and strategies and properties of the firm going into the crisis shaping responses and decisions that are made during the crisis, and this will affect, the human capital and the labor market in a major way. So we're trying to get extra resolution on the decision-making processes within the firm. So I'm from a business school, so I guess that's to be expected.
Yes, thank you. Erling, would you like to add to that? You are muted, please. Could you unmute yourself?
There are obvious complementarities here. And we do study the same problem in some sense. It's, what's the consequences of this pandemic and how is it going to affect the future? But I think we do work with different data, and we do work with different. So I think we will have a sufficient overlap, not overlap. To add to the total picture in a good way, and we will surely keep contact during this research endeavor. So we will just learn from each other. I think we have a focus starting out with our job vacancy things, and then we will merge these things to firm. We will merge data on job creation and job destruction and so on to firm specific characteristics in 3 countries. So that's
yes, thank you. Simen, do you see any connections that will be useful?
Well, I think there's a lot of similarities and connections, and as Lasse said, I think that's just natural, sort of in this phase when we present very, very broad sort of questions or topics during 3 minutes. When you then go back and actually do the research, you will typically sort of narrow it down much more, and we will also take some considerations into what other researchers do and sort of try to adjust the projects such that the overlap will probably be much smaller in practice than what it seems at this stage. Yeah. And if there is some overlap, well, perhaps that's just fine. That will also sort of add to the credibility of research if it actually do find similar results. So.
Thank you. So let's go a bit to the different topics. Lasse, for you, this pandemic has presented us with the perfect storm in a way. We have three shocks occurring at the same time, right? We have the demand side shock, we have the supply side shock, and we have the health crisis. And it's global. It's not only us, and it's occurring under different economical regimes and different ways of handling different impulses to handle things. And this is decidedly not a usual situation. And I'm wondering, how do you treat this combination of underlying shocks and effects in your project to make the outcomes also valid in a more usual setting, which we hope to return to?
Yeah. Well, a famous economist once said that every crisis is different and every crisis is the same. And I think what he meant was that, you know, the causes and the detailed dynamics is unique every time, but there are some patterns that are important across crisis. Of course, you know, we need to understand demand shocks better. We've been studying demand shocks since biblical times. We've been focusing on why the economy suddenly contracts, and there will be demand shocks in the future. We've been studying supply shocks. They're not quite as common, but we've been studying supply shocks, and this will add to our understanding of supply shocks. And of course, it's a medical core of this crisis, but you know, it transforms into demand and supply effects and changes, so to speak. And I think this will add to our library of understanding demand side shocks, supply side shocks, and it will make us even better able to understand the next shock, whether it has a medical problem at the core or whether it's a new shock. It will just deepen our understanding of the kind of situation we're experiencing here, even if the next crisis surely won't be exactly like this one.
Thank you. Then to Erling, as is often the case in a crisis, groups at the margins like youths, more lower qualified persons, and also effects on inequality and polarization as you study, those groups seems to pay a steeper price than the rest of us. Would you think that the obvious need to innovate and digitalize adds or attracts from such effects?
Yeah, maybe I can combine the question with from Oddveig Åsheim
yes,
at the same time, because I mean young people are more hit by recessions than other people because they are entrance to the labor market, and the hires is one of the things that stops quicker than fires. But in this crisis, there's an added component that the young people work to a larger extent in contact jobs that are in contact with other people. So this pandemic crisis we have now is more severe for young people than ordinary crisis. Is my guess. Is my okay. So I think that's one reason why young people are hit even more. Of course, these crises have a tendency to speed up technological transitions. We've seen this before, but there are some tendencies of larger transformations in occupational demand for different types of occupations and so on, following or during a recovery from other crises. And this crisis is special because it's likely to enhance, speed up technological change due to digitalization and automatization because of the nature of the crisis and the fact that we are sitting here now working virtually. And of course, on the one hand, we know that digitalization and automation increases the demand for high-skilled worker and the professionals and engineers and so on, and is to the disadvantage of low-skilled workers in many firms that where such changes occur. But it's also the case that a lot of the jobs traditionally that are not that young people are in are not as much affected by technology because they cannot be automated. For instance, care jobs, hotels and restaurants and things like this that traditionally are less affected by automation. For that reason, we would expect maybe that it wouldn't be such a big problem for young people. But I think one thing that's special in this crisis, following is that we should expect to see a boost in the decline of jobs in retail, and retail turns out to be a very important employer for young people. It's an important job, first job for people when they come out of school. And it's an important job for students, and it's an important job for people with less formal qualifications. And it's huge in terms of magnitude. These are lots of jobs, and a decline in these types of occupations will have a big impact, I think. So we don't know if digitalization. I think it's not clear what we should expect, but there will be huge effects, I think, on the occupational distribution going forward.
Thank you very much. A question for Simon. You mentioned fair burden sharing and social insurance, which from different angles are also touched upon by the other two projects. As we've seen time and again over the course of the pandemic, what serves us as a society is, and what is considered fair on the individual level, do not always coincide, like national restrictions and individual consequences. Any thoughts on how to reflect this in advice to policymakers?
Well, when you design social insurance policies, there's sort of also always a tension between providing insurance on one hand, sort of helping those unfortunately hit by the corona, and providing incentives, sort of making it worthwhile to search for or take a new job. And during this crisis, this tension is sort of strengthened much more as we have extended public insurance programs to also cover firms, not just workers, with policies for cost compensation and wage subsidies, for example. And I think while sort of unemployment insurance has broad support, insurance programs for businesses are much more controversial for good reasons, I think. But it's sort of too early to say whether this was wise or not. I think that is sort of one of the major policy questions we want to evaluate in the aftermath of this crisis. But we sort of first have to study the recovery, because the motivation for these firm insurance policies is that the recovery is going to be faster. And we haven't seen it yet, so it's too early to say. What we have seen is that the number of bankruptcies is much lower than normally, much lower. But again, it's a bit hard to say why. We don't know whether this is because of the policies or other reasons. So yeah, have to wait and see, I think.
Thank you. So let's round off with a reflection on the real-time data. You are combining registered data and real-time data, and I think for the medical research, it's kind of obvious what the real-time effect is to quash the pandemic and help us get through it as soon as possible. But on the economical consequences, what would you say are the new avenues or the new options, possibilities that opens when you get available real-time data together with the registered data? I'd like to pass that to all of you. Let's start with Lasse, for instance.
I think we had a big project here at the Norwegian School of Economics on the financial crisis, and we've studied other recessions and crises before. And usually you have to do that, looking back from after the crisis. You sort of try to look back into the conditions that were there before or during the crisis. So it's retrospective analysis, which has its problems. Now, this time we can understand the dynamics a lot better. For example, we try to, in our project, we try to measure expectations, how expectations and optimism and pessimism among managers change, and how this affects their behavior and decision making, which is important. And so there are opportunities to understand details of the dynamics of the crisis that we, at least to my knowledge, haven't really had at this level before.
Erling, would you like to add to that?
Well, just a brief comment. I think that using all data is very good for research because we know a lot of things, and the data have been studied a bit, and we can study long-term consequences. On the other hand, real-time data gives us a chance to be much more policy relevant, because there's always a new, the world is always different when you have a new reform or crisis or thing or policy initiative. So the more, I think, in terms of impact and in terms of being able to address pertinent policy questions, real-time data is excellent, and we have seen now that it can be used much more, that it can be produced much more effectively, and it can be used quite efficient. But it's a trade-off in terms, because we get to focus on what we can measure quickly, and we don't get the long-term consequences and things that are measured more carefully.
Simon, short comment.
Yeah. I just want to add that some of these real-time data sources, they are actually registered data. So some of these registries, they are updated continuously. So when you file an application for UI benefits, this is actually being registered data at NAV in the same moment. So it's possible to get hold of these data much faster than what we traditionally have done. So I think that's sort of a possibility that we have just started on, and which is highly relevant, perhaps not for research mostly, but much more relevant for policy making and for sort of providing policymakers and the public with updated information. It's hard to sort of produce high quality research in real time. I think that's a very sort of unrealistic ambition.
Yeah, sure. Thank you very much to all of you for taking part in this discussion, and we'll have to save the remaining questions from the chat for the parallel session coming. So this concludes the plenary session, and now it's time for the 3 parallel sessions. And until now, you've all been sitting quietly and listening, and in the parallel sessions, we encourage you all to be active. Please have your cameras on, ask questions, contribute, and say if you can help or even ask for help. You'll be surprised. We're also grateful if you will all spend some minutes afterwards to help us improve to the next seminar. You will all receive an email with an evaluation form.
On behalf of the Research Council, the Cancer Society, and Trond Mohn Foundation, thank you for participating. Best of luck with the parallel sessions, and we hope to see you all back for the 3rd webinar that we have planned for September.